Hello, welcome back to another stream. It has been a long time, it's not like it's only been 5 minutes since the last stream happened, of course. So now that we are here, I would like to show that we have a horizontal chat now. Whoop whoop. So guess what? That'll be very useful for certain types of streams. Karaoke stream. Yeah. At some point, once I have the proper, what's this called, the schedule for it, it might happen. Oh my gosh, wait, the scrolling is being covered. There you go. So yeah, introductions. Hi, I'm Millennium, Isekai'd Masked Knight. Again. Hi, I'm Millennium, Isekai'd Masked Knight Empress Scientist, and today we did Power World and it just ended about 10 minutes ago, and now we're doing science, because you know what, why not? Let's go. And today it will be bioinformatics. But before that even happens, guess what? We are, like, remember one of the posts I said, the 12,000 subscribers celebration, and we did, so let me write it just to explain it. So we whoa, that's too big. 1 second, let me erase. So it's 100, it's 12,000 subs, right? To celebrate, this is the whoop whoop. I know. Now here's the thing, I was like, oh, why not do 1,200 not push ups, oh my gosh, jumping jacks. And I was like, I'll do 120 per stream. Not necessarily straight, because it depends on the situation. For example, if I just ate, I will not do it. So we finished day 1 of it and day 2, and we are now at 120 plus 120. We have 240 jumping jacks now. And I'll do 120 right now too. So here it goes. Let me just check. Is the heart rate monitor working? It has to work, because that's the proof of purchase. That's the recibo. So let's go. The mic might be far away because I'm doing the jumps. Alright, let's go. Wah. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Let me breathe. 1 second. That was 30. Take note. Yep, there you go. Okay. Couple more. So that was 30. I have to remember. And 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50. You know, it's pretty tiring. Now we're at 50. This will lengthen the lifespan by 1 year per jump. 50. Wow. Look at the heart rate go. Okay. Let me adjust the track. There you go. And 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, 66. Boy, 진짜. Kuya ni. Wait lang. 66. I think in the In K-pop terms, they would say him, de, ro. 66. Okay. Stretch a little bit. And let's go. 7, 68, 69, 70, 71, 72, 73, 74, 75, 76, 77, 78, 79, 80. Wah! Malayo pa! 80. Still too far. One moment. Okay, 40 more. Fighting, and then we'll discuss bioinformatics. 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, 93, 94, 95. Calma! 95. Breathe. Breathe, breathe, breathe. 95. And let's go. 96, 97, 98, 99, 100, 1, 2, 103, 104, 105. Wait lang. We are at 105. Almost. 106, 107, 108, 109, 110. Last. 10. Whoa. Okay. And then 111, 112, 113, 114, 115, 116, 117, 118, 119, 120. We did it! Whoa. And it is finished. Imagine if I just ended the stream there. Alright, so now that we did 120, the grand total is now 360. Whoop whoop. So that's the update. And now on to bioinformatics. Let's go. Okay, now what is bioinformatics? It is, wait, let me write it. Bioinformatics. So it involves the development or application of computational tools and approaches to understand different things, you know, like, for example, what's this called? Biological, medical, behavioral, and health data. So that's what it is. And in the context of just genetics and genomics, you have it provides methods to assemble or sequence it. For example, your molecules like the DNA and RNA. And scientists depend on bioinformatics to study like the gene function, genetic variation, the molecular basis of the diseases. So that's why we need bioinformatics. AKA, what is it? It's using computational tools to study the different sequencing and all that jazz. So the application for it, bioinformatics applications span almost every branch of medical science. So for example, from the basic research, wait, I forgot to check, does the mic work right now? Let me see. Yeah, it does, it works. Yeah. So bioinformatics span nearly almost a gazillion, all of the branch of medical science. And you have from basic research like genomics, transcriptomics, proteomics, until you have the clinical fields like oncology, cardiology, infectious diseases. It would support understanding those specific disease mechanisms. And take note, as they become cheaper and faster, it's not, you can then use them for the different disciplines. So essentially, if they become cheaper and more efficient, the usage of it will expand. Okay, it will expand. The horizontal chat looks really nice. What we'll be able to use that one day for a possible karaoke. So what generates big data? Okay, biological big data would be from very large scale experiments such as genome, exome, and transcriptome sequencing. And you can also get them from RNA-seq, ATAC-seq, ChIP-seq, and other things. So you can get a lot of data from those. And how does the workflow work? I have to draw this one. It's an interesting flow. Here. The way it works. Okay, there. Wait, is the fan on? Yeah. The way it works would be you have your samples or control / experimental. And then you have your DNA, your RNA, your single cell, your chromatin, and then you have the proteins, and then you have histology. After that, you can make the sequencing libraries over here. I'll just write it this way: sequencing library. And then you sequence it, and then after that you have the different methods like the mass spec, the FACS, and qPCR or RT-qPCR. But that's essentially what the general workflow is. And as for what the sequencing does, it would be for a sequencing by synthesis method, which would be like adding the nucleotides, or you detect the current nucleotides there. Because as you make your DNA, the fluorescently labeled nucleotides, aka the nucleotides that are like shiny, they're added to those, and those are the ones detected. And then you generate the short reads for it, and those raw data is generated or stored initially in FASTQ files. So that's how it works. So again, as they sparkle like shinies, they would then be read and detected and stored as FASTQ, aka this is how it's spelled, FASTQ files. So if you've ever tried or heard of the terms in RNA sequencing, you'd really hear FASTQ. And we mentioned the workflow. Okay, how much sequencing for the human genome? The human genome is about 3 gigabases in size. And to achieve the 30 times coverage, you would need about 90 GB of data per genome. That's a lot. And again, FASTQ files contain the sequencing reads. And there are some pictures of FASTQ files, but essentially it has a lot of letters. Just remember that it has a lot of letters. There's also terms called the SAM, the BAM, and the CRAM files. Just it contains the mapped and unmapped sequencing reads, and they're very compressed. Let me write it down. The BAM. Here. Write it over here. The BAM, the SAM. Nope. The order is wrong. Just remember the SAM, the BAM, and the CRAM. The other say I search for images of DNA, compare the size of atoms, and DNA is tiny. It's only a few atoms across. Yes, it's very, very, very tiny. So imagine, oh, so that's how the horizontal one works. Okay, yeah. So imagine the how coiled the DNA is for us to be able to see it as a chromosome, like as the X. So it's very the very very small, the very original version before you coil it so much. It's very very tiny. And we talked about the BAM exam. That's all right. Oh, it has clinical. No, it's fine. Mutation examples. You can use like the UCSC genome browser, Golden Helix genome browser. Just different sites, but one of the number one sites to remember would be the NCBI site. This one's nope, N, not M. One of the most important ones would be the NCBI site. That one. Don't forget that. It's a very important website. And then the second website would be UCSC. That one. Those are some important sites you would need if you really want to dive deeper. UCSC. Okay, because this browser would be like, it's a genome browser, and if you open this one, if you search it, it would have a big picture of the DNA like that at the left side, and it would have like R tools and stuff like that. And then oh, the sequencing analysis workflow. What is the sequencing analysis workflow? Very very good question. That one is, you have the sample prep, the library prep, and sequencing, plus primer analysis, like the QC filter, secondary analysis, and tertiary. There you go. So just, it has a flow. You prepare your sample, then you do the library, then you sequence, then you analyze. Again, sample prep, library prep, sequencing, analyze. Okay, good. Alignment. Oh, and the third website to remember is GO, the Gene Ontology website. That one's a funny one because you can see a lot of things. And that one, nope. Okay, so question time. What is bioinformatics? It is the body of science. Bioinformatics is the development or application of computational tools to analyze biological, medical, or health data. Again, bioinformatics is the application of computational tools to analyze health data. In what areas of medicine is bioinformatics used? In all of them, a lot of them, like genomics, transcriptomics, oncology, cardiology, infection, diagnostics, personalized medicine. What types of biological experiments generate big data? RNA-seq, ATAC-seq, ChIP-seq. Workflow is sample collection, or DNA, RNA, protein extraction, labeling, hybridize. How does Illumina sequencing work? DNA is synthesized. You base by base, and then you add the fluorescently labeled nucleotides. SAM, BAM, and CRAM. That's in order of from not compressed to super compressed. SAM is uncompressed. BAM is binary compressed SAM file. CRAM is very, very compressed CRAM. Okay? What is contained in a FASTQ file? 4 lines, header, sequence, separator, and quality score. And how does single-cell RNA-seq differ from bulk-seq? Well, single-cell would be for individual cells, while bulk-seq would be like, it will miss a lot of things. Therefore, single-seq is, I think it's better. because it would reveal some differences that's missed out on the book. Oh, not the taxi. That's fine. Oh, if you see differentially expressed genes clustered around Wnt5A, this is a very specific question. What does it suggest? It would suggest that in this specific disease condition, the Wnt pathway might be dysfunctional or dysregulated. So yeah, guess what? The biggest plot twist is we finished bioinformatics. And I'll move on to discussing genetics and epigenetics now. I'll just update the thumbnail later. Erase. So now we are discussing genetics and epigenetics. Whoop whoop. Okay, that's crazy. So, for genetics, you have, this is just a very specific and small intro to it. So you have the key statistical concepts, right? That's important to understand the genetic variation. And then just as a refresher, what is a random variable? It's a quantity. Wait, the heart rate monitor stopped. Let me just reset it real quick. And here. Did it work? Does it work now? Is it working? I think it should be working. It should, yeah. So again, what is statistics? It's a random variable. This is a statistics refresher. It's a random variable. A random variable is a quantity that can take multiple possible values, okay? And each is associated with a probability. The expectation or mean or variance, the spread, would describe the average outcome of it. And that's why the random variables would extend some basic algebraic variables. So in algebra, you have things like 3x equals 9, which has a single correct solution, right? aka x equals 3, for example, because 3 times 3 equals 9. Voila. But in random variables, you could have different multiple values, so you would have like a set or something like that. And then you have the binomial variable also, but yeah, it's fine. Next, genetic architecture. What is genetic architecture? It would describe which genetic variants are associated with a trait, but the most important part is how many variants contribute and how they interact with the environment and how those effects are distributed across the population data. So the question now I want to ask, how many variants? Okay, here's the thing, at least one must influence a trait phenotype so that you can see something, but the number can be infinite. Like Gojo. Infinite. Okay, so Mendelian, it's monogenic. A single gene influences the trait. You also have polygenic, aka countless genes contribute to it. And then you have oligogenic, a small number of genes. Then you have monogenic traits, which are traits controlled by a single gene. And you have the law of independent segregation, aka siblings. If you have siblings, during meiosis, the sibling pairs share, on average, 50% of their genome and have a 50% chance of sharing a very particular allele. And that law is strictly true when alleles are not linked. And Mendelian inheritance would describe traits with about 100% inheritability. It would follow the dominant and recessive patterns as well. So yeah. Okay, so here's the thing. The human height, how tall you are, it does not follow a simple Mendelian inheritance pattern. I think this might be important, so stay tuned. Because it's that height is influenced by many genes. So the question now is it is it 2, 3, or 4, or an infinite number of genes contributing to the low side? And notably, around 1918, wait, I have to write this. Around 1918, someone raised the idea that traits like height arise from an action of countless genes contributing to the small effects. So that's why it's a shift from the Mendelian genetics that we know. Okay, so and there's let me talk about the sibling heights and all of that. Here you go. Okay, so you have something called the infinitesimal polygenic model, okay, and mid-parent concept. So if one sibling is taller than 99% of the other people, how tall do we expect the next sibling is? Okay, so the infinitesimal polygenic model it has been used for about 150 years. It provides a framework, and it would introduce the concept of the mid-parent value over there. So an average trait value of the two parents would predict the distribution of their offspring's traits. So there's a within family variation as well, but it remains stable across the unrelated families. All right. Yeah. There we go. So, going back, what basic statistical and biological knowledge would be necessary for these? You would need basic statistics, the central limit theorem, the law of large numbers, and genetics concepts like meiosis, heritability, and polygenic. And what is a random variable, and how does it differ from the algebraic variable? The random variable would be able to take multiple values within certain probabilities, but your algebraic variable only has one fixed solution. And what are the first and second moments? The first moment is the mean. Second moment is the variance or the spread. They're crucial. Make sure you don't mix them up. What's the Bernoulli random variable? Related to probabilities. What defines the binomial random variable? It is the number of successes in the n independent Bernoulli trials. Whoop whoop. Okay, what's the central limit theorem? This is a good one. Erase. The central limit theorem. Central limit theorem. That one would state that the sum of many independent random variables approximates a normal distribution. So that's why it would explain complex traits like the height, so that it follows a normal distribution. And what are the three categories of traits? That's the monogenic, oligogenic, polygenic, single gene, few, and many genes. And what does the two allele dominant model demonstrate? It would show how, hello cheeseburger, it's been a while since I last streamed, aka 10 minutes ago, since the last Pal World one. And now we are yapping about, we just finished bioinformatics and we are now at genetics actually. So the next question now is, what is the law of independent segregation in meiosis? During meiosis, Each pair of alleles segregates independently, which would then give the siblings a 50% chance of sharing any given allele. So you share a ton with your siblings. Okay? What's the two allele dominant model? It would show how multiple alleles with different effects and environmental factors, how those combine to shape trait expression. Okay? Oh, and I can see the different shields. I think one I can see now, I think in Rojas would be 2 swords now, and because I think that would be 2 months or something. And then Cheeseburger has the 1 sword. So whoop whoop there, so it actually works. The badges work. The designs upgrade after a few months. Yeah. Yes. Oh, CRISPR clustered regularly interspaced short palindromic repeats. Okay. Oh, and why is there no single height gene? Why can't you just be tall if you want to be tall? Well, the height is a polygenic trait, so it depends on maybe a couple of genes or thousands of genes, each with a small effect, not just one. And oh, we already talked about this one. Last, what evolutionary forces affect sibling similarities? Yes, we said the thing. We said the CRISPR. What evolutionary forces affect sibling similarities? Well, there is the stabilization selection, aka you keep the traits optimal. And there is also directional shifting traits, and you have divergent selection, aka the splitting of traits in the population. How would you test whether height follows the infinitesimal model? You analyze a lot of families. You analyze a large family of cohorts and check if the height shows normal distribution. Okay? Okay. Now, how would you design an experiment to test or estimate how heritable the trait is? So you measure the trait in different siblings and calculate the mid-parent averages. And lastly, how could conditional sibling distributions help distinguish between polygenic and rare variant models? Okay, in a polygenic model, the sibling traits should be correlated, but rare variant models would predict the extreme differences if only one sibling carry that mutation. Essentially, if your sibling is extremely tall, there's a chance you might be tall or you might be short. It just depends. But that is bioinformatics and genetics. Yeah, thank you for coming by to this mini chat after the Power World, the mini science. Yeah, yep. Thank you for watching, and I was gonna say sayonara. No, see you next time. Bye.